And yeah, so that just signals that the main talk will be recorded, and then the recording will stop before we get into the Q&A, so you can feel disinhibited to ask all the questions you want to ask of Jaan. Okay, so it's my great pleasure today to introduce to you Jaan Aru. So Jaan did his master's at the Berlin Humboldt University and then did his PhD research at the Max Planck Institute for Brain Research under the supervision of Lucia Maloney and Wolf Singer. And he then moved back to Estonia, where he's from, to take up a postdoctoral position in Tallis Batman's lab, and then he'd now become a associate professor at the University of Tartu in Estonia. And many of you will know Jaan from his work on trying to provide a taxonomy for different types of neural correlates of consciousness, such as the pre-NCC and the post-NCC. And more recently, he's now been getting interested in doing work on cellular mechanisms of consciousness, which I think is something that's quite novel to our group. We've tended to have talks on the more cognitive level, but I think this is a super interesting topic and very important for our field. So it's a real pleasure to have you speak to us, Jaan, and take it away.
Thank you very much. It's a pleasure to be here. And, you know, I know my CV is a bit difficult to find even online. So you missed a crucial point there, which is that actually my last postdoc I did with Matthew Lacum in Berlin. And that's where actually this material for this talk is coming from. But no worries, I will clarify that as
Apologies,
we go.
I was trying to search the website.
No worries. No, it's I even didn't have a homepage until you guys asked whether I have a homepage or something. Then I made quickly something. I'm not a fan of these things, so I try to do science. But let me try to do this talk now. Okay, so I think we all kind of know and agree that one of the starting points of the modern science of consciousness was this influential paper by Francis Crick and Christoph Koch. And here I just want to point out the title of this paper, which is Towards a Neurobiological Theory of Consciousness. So you see that here in this title, they have this word neurobiology, right? And if you go and read their subsequent papers, for example, here's from 1992 piece, we need to construct neurobiological theories of visual awareness, etc. They were really focused on this neurobiology. And that was mainly coming from Francis Crick, but of course also from Christoph Koch. And that was very important in the 1990s. But I guess we all kind of agree that this thing, the study of neurobiology, hasn't been so relevant in the last decade, maybe in the last decades, last 2 decades. So it has been relatively unimportant. And it's because many other things have come up, many more interesting topics, etc. However, I feel, and I guess some of you also feel, that it is also a shame, or it is unfortunate, because in the end, one of the key facts about the brain that we know is that somehow consciousness is generated in the brain. So it should be important to try to study it and try to understand how the brain can give rise to consciousness. So here I really mainly want to focus on what we proposed in this 2020 Trends in Cognitive Sciences paper, where we try to put forward such a neurobiological framework. I don't know if we made a good job or not, it's for you to decide, but today I want to tell you about it. And I won't explain this figure now, I just want to put it up here to illustrate that it's a neurobiological framework which includes several cellular mechanisms, and then tries to explain properties of conscious experience. But here's the outline of what I try to do today. So first, I will try to ask this question about what do we actually mean with neurobiology? And then I don't assume that all of you are experts in neurobiology, experts in pyramidal cells and dendrites. So I will cover this part also actually rather thoroughly. And then I will try to say what this neurobiology actually implies for the science of consciousness. If I don't make it through everything, I think it's fine. Today, I just want to convey to you that neurobiology is super interesting, actually, and it can tell us something about consciousness. So very briefly, what do we mean with a neurobiological hypothesis? And here, an example again from the old papers by Koch and Crick. Here they say, thus a very simplistic answer to the question, which neurons fire in such a way that they correlate with awareness would be the large pyramidal cells in layer 5 that fire in bursts and project outside the cortical system. It would be marvelous if this were true. And it was a very, very bold claim in the year 1994. But now, 27 years later, we think that it is actually true, or it might be true. And I find it simply amazing that in the year 1994, they could make this hypothesis. But of course, back then, they didn't have the tools. They didn't have the tools to actually show this, right? But now, neurobiology is equipped with the best tools. I mean, I won't explain it to you, but I guess all of you basically know about optogenetics, right? The idea that you can control neural activity with light. And the key point is that you can control different cell types given you have somehow figured out a genetic marker of that cell type. And through these tools, actually, there is now evidence that what Crick and Koch were proposing is true. So let me tell you about this layer 5 pyramidal cells. Here's a plot from Max Shine's paper, which is a very nice paper. And so here you see the cortex. I hope you see my cursor. I will try to not rely too much on it. But anyway, what you see is that, yeah, you have the cortex. And of course, even in consciousness science, most people are cortical chauvinists, right? They only tell about the cortex. Everything happens, feed-forward feedback, everything happens in cortex. But the truth is that, you know, the cortex by itself, it doesn't have a lot of influences directly on behavior. So it cannot control behavior without certain cell classes. And one of the cell classes is this layer 6 thalamocortical cell, which controls thalamus and has loops with the thalamus. But we will focus on this layer 5 pyramidal cells here on the left, because they are cells that send extensive projections to the stratum and to the thalamus, and even all the way to the brainstem and to pons. So in very simple terms, we could say that if we would ablate all layer 5 pyramidal cells, then the cortex, it can do all these marvelous computations, but it would have zero effect on the behavior. And this fact that these layer 5 pyramidal cells are the ones that are the main output of the cortex was one of the reasons why Francis Crick and Christoph Koch actually proposed that these cells could be crucial for consciousness. So here we have a couple of such cells. And if we now put the cortical layers here, then you see that, yes, they're cell bodies. are at layer 5. That's why they are layer 5 pyramidal cells. But the interesting aspect is that their dendrites go all the way to the surface of the brain, to the layer 1. And actually there they have extensive branching and make extensive connections. So that is very interesting. And that will be one of the key facts for this talk. So I will mainly talk about putting this into 2 compartments, saying that here around layer 1, layer 2, we have the apical compartment. And there, here below at layer 5, we have the basal compartment. And why this is interesting, or why should we put this to these 2 different compartments? Well, one of the most fascinating facts about the cortex is that a lot of the feedback connections go to layer 1. And here you can see it from a plot in a very recent paper by Schumann and others, where they basically, here you have the 6 cortical layers, right? And you have 7 different areas. And in all cases, you are looking at the projections in the rat, mouse somatosensory cortex. And the main point is that you see that other cortical areas, for example, here on the left, and also thalamic areas here on the right, they project heavily to layer 1. Yes, some areas like the POM, the higher of the thalamus, also projects to other bands of the cortex. And yes, there are projections to layer 5 and layer 6, but there's this interesting pattern that all these areas project to layer 1 of the cortex. And usually when it comes to neurobiology, then structure defines function, right? So it's not coincidence that they all project there to layer 1. And it's not coincidence that, you know, the apical dendrites, the dendrites reach layer 1, right? But rather, as Matthew Lacum in Berlin has proposed, this is functional, because these are not simply 2 different compartments of the pyramidal cell, but they are 2 different functional compartments that receive different input that have different functions. And I mean, in his earlier papers, he said that if you could think of this top compartment here as a compartment that receives top-down information, that receives feedback. And the other compartment is the one that we usually think of when we think of neurons, or when we are thinking of artificial neural networks. So these are this is the compartment that receives input from the bottom, from the data, from the world. And this is, of course, in some sense simplistic, but it contains these important facts that, you know, in the brain, we see this segregation. And we see that the apical dendrites go to the layer 1. So it makes sense to think, okay, these are these 2 separate compartments. Now, let's see, what do they do actually? And Matthew Lacum actually became famous for these experiments that were more than 20 years ago published in Nature when he was a postdoc. He was in the lab of a Nobel Prize winner, Bert Sackman, and there they did this super complicated experiment. So they put not 1 electrode into a cell, but 3 different electrodes. So that's super complicated to do in an in vitro setting. And that's why it's a Nature paper. And then if you have this setup, then what you can do is you put a little current here and there. And the logic is that, yeah, now you can actually check what happens if these 2 compartments interact. And so this is what happens. Basically, if you just stimulate here on the top, you see a small activity. If you just stimulate the basal compartment, you see one spike. But if you now combine these two, then you get a burst of spike. So the neuron basically goes crazy, starts firing as hell. So what you did basically is you combined two data streams. And that was, I think, the revelation for Matthew Lacombe also, that, you know, there is a cellular mechanism actually within each pyramidal cell that signals a match between these two streams. And his original hypothesis is put forward in this Trends in Neurosciences paper. And now you see that it becomes a bit more interesting about, what are actually these streams? And you see that now I kind of opened up a bit, right? So I'm trying to convey the message here that, you know, perhaps we don't need to decide what exactly these streams are, but we have to just decide that, look, these are qualitatively different information. So we could call the information reaching the top compartment that these are higher order representations. We could say that it's a model. We could say that it's context or predictions. And to the bottom compartment, the basal compartment, we could say these are prediction errors going there. We could say that it's bottom-up information. But the most important thing is that there is this separation, and basically said in very simple terms, the information that one compartment receives is kind of more boring. And the information that the top compartment receives is super interesting, right? It's context, it's everything else the brain knows about this particular thing. And this was the reason why I became interested in Matthew's work. And you have to think about it. I mean, I was just, I had worked on virtual reality, artificial neural networks, yes, also intracranial recordings, TMS, but nevertheless, I had done human studies. And I will bring one example here just to show you, try to explain to you why I was so interested in Matthew's ideas. And this paper basically illustrates my long question after how can we actually explain this pervasive nature of top-down effects on conscious experience, right? I won't bring any examples because I saw that you had some previous talks that probably brought you a ton of examples about top-down effects, but I want to just, you know, say that this was really important for me, coming from cognitive neuroscience and psychology, to understand the nature of top-down effects and how we can explain it. And just to maybe bring one general example, right? Currently, your eyes are moving, your eyes are blinking, and the image on your retina is shifting. It's changing all the time. But I guess your conscious experience is stable, it's continuous, it's smooth, right? And that is maybe the most pervasive nature or example of the effects of top-down influences. Anyway, I will also bring one more concrete example that I was just finishing by the time I had to think about my next postdoc. And this was this paper that we published in Consciousness and Cognition. And the task goes like that. So basically, it's what you have to do is it's a dual task. So on most trials, you're asked whether the faces have the same orientation or not. And you just have to basically do a discrimination task. And then on a subset of trials, I guess roughly 10% of trials, you're asked about the subjective visibility of this Landolt square. So you do this perceptual awareness scale. And we train the subjects well. So there are previous training sessions also where both tasks are done independently, separately. So they do this very well. And then in the second half of the trials, we do a trick. So we actually present this kind of an image, right? But on these 4 trials, we ask them nevertheless, you know, how well did you perceive the square? And perhaps you would expect that subjects tell you, come on, you're stupid. There was no square. What are you talking about? But that's not what happens. That's not what happens. So actually in this particular experiment, 12 out of 15 subjects at least once used this rating that I had almost clear impression of the square, although there was no square on the screen, right? Yeah, of course, one could say that perhaps this is some kind of a bias and we didn't believe it also in the beginning. But if you talk to the subjects, so after the experiment is done, you talk to the subjects, then you see that they are genuinely surprised to learn that there was no square on the screen. And we have now replicated this in 5 different samples in 3 different kinds of experiments. So it's not always these faces, it's different things too. So we're pretty sure that it's the case. But you know, the question always was, okay, how do we actually explain it? And this was to say it, you know, in a very short to the way that, you know, that's why I was interested in Matthew's work. That's why I actually wanted to go to his lab. And I did. I did go to his lab. And I think, you know, if you think about it, you know, me, a psychologist and cognitive neuroscientist going into Matthew's lab, then it's basically this anecdote that a guy walks into the bar, right? Because I had done experiments with humans. And, you know, to a human, you can tell basically in 3 minutes what he has to do. But now I had to train mice and work with mice. So that was a big, big challenge. But let's say if you kind of agree or find something interesting from this talk today, then all this challenge paid off, right? And so when I went into Matthew's lab, then I talked with Mototaka Suzuki. He is another postdoc in the lab. And at the time, he was completing an experiment. And the experiment went like this. So basically, he shone light on the apical compartment. And he did it in 2 different conditions. So in one condition, the mice were basically awake, conscious. In the other conditions, the mice were anesthetized. And then he shines the same light to the apical dendrites of these neurons. But what he observed is that although the light here on top is exactly the same, what comes out is not the same. So if you now do simultaneous measurements at the cell bodies, then you find that in the conscious state, the activation of these apical dendrites leads to activation of the cells. But in the anesthetized case, nothing comes through. It's as if here's some block, something is happening here that doesn't let this input that arrives at the apical compartment through. So nothing gets through. And I think he was pretty happy about this finding. But then he was genuinely surprised to see how I started jumping around saying, oh my God, I mean, this is the most amazing finding in the science of consciousness. Because, and let's go now back to this image, right? Now, this means that basically all the ideas I presented to you now about 2 compartments and 2 compartments having different information were actually relevant to the question of consciousness. because it turned out that, basically what happens is that during unconsciousness, this functionality or coupling between the two compartments is cut. So that was a finding about consciousness, which to me immediately had amazing implications for the way we think about consciousness. And, so it was a surprising finding, but I think you immediately I immediately understood that this has very direct relevance for how we think about consciousness. Because, and I will explain it now in more detail, basically you could say that consciousness, it turns out, is related to this integration between the top-down information and the bottom-up information. There's some integration happening that's needed for consciousness. If you cut it, you don't have consciousness. And basically, then what we did is we put these implications and these ideas into this paper. And I will try to now rather briefly explain the main implications I think this kind of an idea has. So first of all, and probably you already got it, the point is that, there's this very fundamental difference between the conscious and unconscious state. And kind of surprisingly, actually, it manifests in this decoupling within single pyramidal neurons. So for me, that's amazing, right? That you have a cellular mechanism that basically cuts the two compartments, and that correlates with changes in consciousness. So, and as I mentioned briefly, or maybe I didn't, but then I have the chance here to say that, what they figured out in this paper with Mototaka Suzuki and Matthew Lacum is that, this input comes from higher order thalamus that controls this coupling. So in other terms, this kind of a red line here is controlled by input from higher order thalamus. So there is a cellular mechanism that is relevant for consciousness, and that is controlled by higher order thalamus and through some specific cellular effects. I won't go into them now. If somebody wants, you can ask later. So, or in simplified terms, then this is the main point that, higher order thalamus controls this coupling between the top-down and the bottom-up compartment. And let me go now to the main implications I think this has. So the first implication is that if you think about it, about consciousness like that, then we see that, the state and contents of consciousness are always kind of coupled. And we made that point in an earlier paper, 2019, and we already knew then about Mototaka's findings, but of course these were not published, so we couldn't mention them. So we made this more general point. But already then we were thinking about what this implies, what this means, why this is relevant. And the point is that, when you look into the papers dealing with the content and state of consciousness, then it's a bit simplified, but you could see this dichotomy that contents of consciousness usually are thought to be cortical, right? Basically, cortex computes the contents, and then, the state is related to some thalamic nuclei, brainstem, right? And basically, you could keep them separated. But if you now think that there are these layer 5 pyramidal cells that are within the cortical loop and that also project to the higher order thalamus and are with the thalamic loop, then you see that, these two loops are coupled through this layer 5 pyramidal cells. And therefore, basically, the dichotomy between state and content of consciousness is also not so clear. Or let's put it this way, state and contents of consciousness are intertwined because the processes are intertwined at the level of cortical cells, cortical pyramidal cells. So basically, let's look at the right here. If the pyramidal cells are in the conscious mode, basically they are propagating information, then you could send some information from another cortical area to the apical dendrites, it propagates, it goes to the thalamus, it completes the loop. But in the non-conscious case, this does not happen. And that explains why basically you cannot have the situation that you are unconscious in an unconscious state, but you have some conscious contents, right? Because they are coupled neurobiologically. But that's not maybe the main implication we want to make here. The main implication we want to make in this trends paper is that, what Suzuki and Lacum discovered is a local mechanism. But the influence it has is global. So in short, so far, I have basically told you, look, oh, these are these amazing layer 5 pyramidal cells. They have 2 compartments and they can be cut here in the middle. And this is related to consciousness. But it's important to understand that there are, of course, tens of millions of these pyramidal cells. And this happens in a massively parallel fashion, right? So in fact, we don't know how local you can do this control of controlling different coupling of different pyramidal neurons. But the point is that it is a local mechanism. But actually, the influence it has is a global one. And of course, if I now think about this image, then it's highly simplified, right? Because here we have only 6 of those pyramidal neurons, right? But actually, the point is that when you couple these compartments, then it has a major effect on all the dynamics all over the brain. So basically, when I, for example, look here and take a quote from the amazing paper by Demerzi and others, where they, I think it's in the abstract, say that consciousness rests on the brain's ability to sustain rich brain dynamics. And then what we think is, okay, yes, it is true, but why this is the case? What are the mechanisms that support this rich brain dynamics? And I guess you already see our answer that the brain's ability to sustain this rich brain dynamics rests on this dendritic integration of these 2 streams, right? And this is what I kind of hinted at already before, right? If you are in this mode where the pyramidal cell is coupled, where the signal flows from the top to the bottom, then if you generate some activity, it can flow basically in this circuit. It can engage the thalamus, it can generate loops. But if it's in an unconscious state, then all these kind of connections are weaker. And we could even, we could imagine that there's some activity in the thalamus, right? That maybe generates some activity in the basal compartments. Perhaps even, because it could go through certain thalamic nuclei to the apical compartments, perhaps even activates those apical compartments. But there it stops. That's where it stops, because this information can't get through. It cannot complete the loop. It cannot make this complex sustained dynamics because the loop is cut, and the loop is cut at the level of layer 5 pyramidal neurons. And here, just a reminder again, I think it's maybe hard to keep in mind all of this, I apologize for that. That, you know, it's not simply, we're not simply talking about the kind of anatomical decoupling, but we're also saying that, you know, this is functional decoupling. This is a decoupling between the top-down information, between the context and the bottom-up information. So it also has functional implications. And when we look now into the conscious state, then in the conscious state, all these loops kind of work. And what can happen then is that you basically, some loops get into recurrent mode, they get stronger, and that could, for example, maintain working memory. It could underlie some other functions, but the point is that in the conscious state, this is possible because there is this connection within the pyramidal cell. There is this coupling between the two compartments, and this underlies this rich, complex dynamics. And I mean, now I probably don't have to explain it. I mean, you immediately see that this kind of model can easily explain also the amazing findings by Marcello Massimini and others, where they show that in the wakeful condition, TMS leads to a complex activity pattern, whereas in NREM sleep or anesthesia or the disorders of consciousness, there's only this local activation. As I showed you some slides ago, I mean, the basal compartments can be activated, the thalamus can be activated, but the loop, it does not propagate. Activity does not flow, it does not propagate, it does not lead to subsequent activation. So that is the second main point we think we make and try to make in this paper, that you know, you can have this local mechanism, but it actually controls the global dynamics. So this local cellular mechanism can explain the differences in global dynamics between the conscious and the unconscious states. Now, the most important implication for us is that this dendritic integration of top-down and bottom-up information streams is the essence of conscious processing. And I think it's tempting to immediately say, hey, but, you know, in unconscious state, you might also have, you know, combination of priors with feed-forward information. So it's not that having modulation through priors is specific to conscious state, but we're here actually making the opposite claim. We're saying that conscious experience is always modulated by top-down information. There's nothing like pure data in consciousness, and that this is one of the main implications of this theory, actually. To put it like this, right, that the fact that, you know, you have these two compartments coupled is crucial for consciousness, and it immediately also has functional implications, because it says that, as in conscious states, you have this integration between these two compartments. In conscious state, processing is always also modulated by the top-down compartment. And I think for me, what's important, and I hope I can convince you also, is that the cellular mechanism is interesting because it naturally explains why conscious experience is kind of has all these effects by priors and predictions, right? What I mean is that it is within the theory. So it is the mechanism itself that already tells you that, look, consciousness is modulated by prior knowledge. So we don't have to make any extra assumptions to explain these massive effects of predictions or prior knowledge on consciousness. We have it in the theory, or that is the theory. Whereas when you would, for example, take here two other very important theories and the two leading theories, right, then of course the IIT and the global neural workspace theory, they can explain these effects of priors on perception. But for that, they have to make an extra assumption, or they have to kind of do something extra. So it's not inherent in their theories. And I'm not saying that this is a big problem for these theories. I'm just trying to point out that first, as you all know, that our conscious experience is affected by prior knowledge. And second, that at least our idea or the cellular mechanism we propose very naturally explains this. And now I think at this point, you might want to ask again, hey, what are these streams actually? Okay, so you always go back to saying that these are these two streams and you use these different words for it. So here again, you see different words for these different streams. And I think this was one of the big revelations when we were writing up this Trends paper, that, you know, we don't need to say explicitly what these streams are, because of course different people coming from different kind of context and background, they will have their own idea about what this might be or how you actually call it. But rather, it is really central for us that there are two qualitatively different input streams. Different people can put different names to them. Perhaps even we will figure out that, you know, maybe the input to apical dendrites from thalamus does some kind of modulation. The input from higher cortical areas does prediction. The input from lateral connections does some kind of like a context. But the heart of the matter is that there are two different compartments and that they kind of do different things. So I apologize if this seems vague, but it is vague on purpose, because I think that neurobiology tells us that there have to be these two different streams, and we can find these exact names or how we exactly call them also later. The important point is there are two streams, and the integration between them is what makes conscious states, and that shows that conscious states are always modulated through this prior knowledge, top-down context, higher order representations, as you wish. And maybe to be a bit philosophical, one could say that the conscious processing always relies on combining the external information with the internal information. And just to briefly also go to this one case that I promised in my abstract, in certain circumstances, then it can be that the internal excitation, the internal drive controls this processing, even without input from the outside. And this is, for example, the case I was presenting in our study. And this is the case in what Nadine has been studying in mental imagery. For example, where, you know, the task is to imagine something, and this means that internally you have to be driving somehow these cells. But the example I wanted to spend a bit of time is dreaming, because we actually, relatively in parallel, we worked on another paper that is out in Neuroscience Biobehavioral Reviews, where we point out that, you know, perhaps all this story is actually telling us also something about dreaming. And I won't spend too much time here. I just want to point out that the idea is simple, that, you know, well, if you have this top-down information reaching the apical compartment, then perhaps in certain circumstances, you know, it could really basically do it without any input from this basal compartment. And the curious thing about REM sleep is that the REM sleep is, of course, associated with high acetylcholine levels. And recent years, there have been several papers showing that when you increase acetylcholine levels, then you increase this apical drive, you increase the influence of the apical compartment to the basal compartment. And that is, in short, the essence of our DREAMS paper. The DREAMS paper is quite much longer than the 3 slides here, but I want to move on. So I'm just putting out now this kind of a full idea that we can characterize different states of consciousness by saying that, look, the cellular mechanism works kind of differently. This is our conscious state. This is where it kind of propagates nicely. Both compartments contribute. During dreams, the top-down compartment, at least in some areas, is the one that drives activity. And when you're unconscious, then these two compartments are cut off from each other. So this is, I think it's a beautiful story, but I mean, you have to tell me whether you also think it's beautiful. But I will go to the fourth implication, and that's the last implication, and ask, what does it all mean for theories of consciousness? Does it mean anything? And of course, it's possible that everybody will simply ignore what we have been doing. That's life, that's science. But if one would take it seriously, one would think that this is interesting, then I think there's no kind of a conflict between what we are saying and what these two major theories, for example, are proposing. Because when you think about the IIT, then yeah, it says that, information integration is really important in the brain, and in machines, in all kinds of things, right? But the question, for example, that I had, and that Matthew also had, is, okay, but what is the mechanism? How you can control the level of information integration in the brain? And the same about global neural workspace theory, right? It's a nice theory, but what does it actually mean that you now incorporate some local processor into the global workspace? How does that happen neurobiologically? And here the point is that, we don't think that what we're proposing is in conflict with these theories, but rather we think that, it helps to understand that each of the neurons in these theories that are central for IIT or global neural workspace theory actually have these two compartments. Because then you have a very nice mechanism for actually basically modulating, for example, information integration, because you could simply have a mechanism that cuts the cells in the middle functionally, right? And similarly, you could have a mechanism that basically takes some activity pattern and propagates it to the global neural workspace of the brain. Simply, if you activate the neurons through apical compartments, and then it gets into this recurrent loop. But I think even more important for us is to say that, look, actually, you don't have to start from information theory, or you don't have to start from a computational theory if you want to understand consciousness. But rather, you can actually start from the cellular level. You can start from the neurobiology and then put it all together. And if you do that, you might understand the brain in a much better fashion, and you might understand consciousness in another light. So I'm almost at the end. Let me here provide a brief outlook. And I think I won't go into these issues. I just want to say that the big kind of advantage of having a neurobiological hypothesis is that you can actually go and do these studies right now. Goes, for example, the question of whether the same thing happens in deep sleep is a very relevant one, right? So Suzuki and Lacum showed it for anesthesia, but the same mechanism should work for deep sleep. This is doable. I know people are already trying to do it. So, but the point is, you have a very concrete prediction which you can test. As we proposed in our apical drive paper, we should say that, you know, if the animal is in REM sleep, then actually you have to have this propagation, etc. So I won't go into all of these, but I'm saying also that what we are interested in is, you know, okay, all of this is kind of on the level of cells, and it has been figured out in mice, but what can we do with non-invasive methods about it? Can we manipulate it somehow non-invasively? Can we measure it, for example, with EEG, for example, with fMRI? And I do think that this is possible. We have to do a couple of extra steps here. I'm always happy to talk to anyone who is interested in doing that, because the problem in neuroscience, but also in consciousness science, has been that, you know, there's very little crosstalk between the cognitive side, between the cognitive neuroscience side and the more serious neuroscience people. And it's hard to do. I saw it when I went to Matthew's lab, but I think that now this link has been done. This link is there, and everyone is welcome to basically use it, to use this fact that we have now people who understand both the cognitive neuroscience level and the cellular level, and every one of them is actually willing to figure out how the brain works and how the brain really generates consciousness. Thank you very much, and thanks to these people with whom I did these two papers. Thank you very much.
Fantastic. Well, thank you, Jaan, for a wonderful presentation. So first of all, if we could just unmute ourselves and give you a round of applause. Thank you very much.
Well, it does sound better with live audience, I must say.
Yeah, no, I know. We should have a track that we can so we should also stop the recording, right, Nadina? So then, and we're going to use the raise hand